Hi, this is Dr. Jagadish Narendra Prasad, Senior Lecturer in Botany. Today, we are going to discuss chapter at a glance about the chapter Plant Growth and Development. In this chapter, we are going to discuss first how many questions have appeared in the previous NEETs. Let us take from 2023 to 2025. So approximately 3 questions have appeared every year except in 2025. Look at here. In 2023, 3 questions have come. 2024 also there were 3 questions, and 2025 there were 2 questions. Approximately almost in every NEET 3 or 2 questions were appearing. Now let us look at a glance about the various topics under which the questions have appeared in the previous NEETs. See, in most of the NEET exam, like from 2023 to 2025, in most of the exams, questions have mainly appeared from de-differentiation, differentiation and redifferentiation and plant growth regulators, as well as some questions also appeared in the introductory part of the growth. Now look at here. In 2023, there was one question from differentiation, de-differentiation and redifferentiation, and also 2 questions from plant growth regulators. Whereas in 2024, one question from growth, particularly from again from de-differentiation, redifferentiation and differentiation, and 2 questions have come from plant growth regulators. Whereas in 2025, there is 2 questions, and the 2 questions have come from plant growth regulators itself. So it doesn't mean that like from growth rates questions will not appear. Even from the growth rates in the previous NEETs there were questions. So what I mean to say is, tell you is, you have to cover the whole chapter and every line of the NCERT book. Now look at important and tricky lines of the NCERT book. Here one important line most of the students they make mistake is, there is a one important NCERT line you can look into here. One single maize root apical meristem can give rise to more than 17,500 new cells per hour. So it means that they are not saying that one cell will become 17,500 new cells. So what they are saying in this sentence is, meristem, a single meristematic tissue gives 17,500 new cells. Similarly, growth is also measured in terms of cell number. For that they have given the example as watermelon. In watermelon, the fruit grows up to 350,000 times. So length in growth is measured by using pollen tube length as a measurement. And surface area is the best example to know the area, growth in area wise. Similarly, growth also has different phases. There are 3 main phases in the whole process of growth. One of that is meristematic phase, and second one is elongation phase, and third one is maturation phase. I think you are all aware about this diagram. This diagram is so important. Most of them don't read the description that they have given just below the diagram. Look into here, parallel line technique. This diagram is based on parallel line technique. This, don't forget it. We have come across this particular sentence in many of our internal exams. So this is one important thing we have to know. And meristematic, whenever you read this particular phases of growth, just remember about zones of root. Like in zones of root, in the root system, there is a apical meristem, root apical meristem, zone of elongation, zone of maturation. All these phases are there, isn't it? So just remember the zones of root, and by taking that into the mind, you read this particular paragraph so that you will get the clarity. In the same way, growth rates. When it comes to the growth rates, growth per unit time itself is called as growth rate. growth per unit time. As we are taking time as a basis to explain the growth here, so this aspect can be used, like we can use mathematical expressions to understand this particular aspect. And we have two important growth rates here. One is arithmetic growth and geometric growth. Now, let us see about the arithmetic growth. In arithmetic growth, after every division, out of the two cells formed, only one cell retains the division capacity and other cell becomes permanent. Whereas here, after every mitotic division, both the daughter cells retain the division capacity. I think you got the clarity. Here, out of the two cells formed from every parental cell, only one cell retains the division capacity, but other cell becomes permanent. Whereas here, both cells retain the capacity of division. That means in this case, growth is constant. Whereas here, growth is exponential. So if you draw a graph for arithmetic growth, you will get a linear curve. Whereas if you draw a graph for exponential growth, you will get sigmoid curve. You can see I have highlighted an important aspect of this arithmetic growth. The best example for this arithmetic growth is root elongating at a constant rate. So I have very clearly told that here in arithmetic growth, growth takes place at a constant rate. Whereas sigmoid curve, you can see like in all organisms, like particularly growing in natural environment, you can see it in all cells, tissues and organs of a plant. And you are getting sigmoid curve, and here you will be finding the exponential rate of growth. Now, if you use mathematical expression, this arithmetic growth can be expressed mathematically by using a simple formula like LT is equals to L0 plus RT. Whereas in arithmetic growth, we are using a mathematical formula like W1 is equals to W0 e power RT. Here, R is most important thing. R can also be called as efficiency index. In this case, R can also be called as efficiency index or relative growth rate. Otherwise, simply we can say that it explains about amount of new plant material produced, or ability of a plant to produce new plant material. That is the importance of this R here. Now look into this diagram once again. If you take a zygote, in the zygote, initially you can find geometric type of growth. Like every cell undergoes division and every cell will have division capacity. Whereas later it turns into arithmetic growth. So in the life of a plant, you can see both geometric growth as well as arithmetic growth. Both are observed. So this is very important. Next, similarly, quantitative comparison of growth of living systems can also be made in two ways. One is absolute growth rate and second one is a relative growth rate. Absolute growth rate is nothing but AGR. That is, you can simply say that expressed growth of a given system per unit time, or simply to say final size minus initial size. That is absolute growth rate. And relative growth rate is one where you are comparing like growth of two different organs or different organisms in a given time. So growth per unit time divided by initial growth into 100 will give you the relative growth rate. Now the most important topic of this chapter, and most of the students are getting confused for finding the examples or concepts related to this particular chapter, is differentiation, dedifferentiation and redifferentiation. I will give you a simple flow chart to understand the entire topic here. Let us look into this flow chart here. So you know that differentiation is nothing but, let us see first the definitions of differentiation, dedifferentiation, then we will go into this flowchart. Differentiation is nothing but, it is a process of maturation that takes place primary meristems. Remember, very simple. It is a process of maturation that takes place in the primary meristems. If primary meristem becomes permanent tissue, it is called differentiation. Under certain conditions, living permanent tissues, if regains division capacity, if they regain the division capacity, then it is called dedifferentiation. Very simple. Here, primary meristem become permanent, whereas here, this permanent tissue once again meristem. What kind of meristem? Such meristems are called as secondary meristems. If this secondary meristem once again loses the division capacity, and again becomes permanent, then it is called redifferentiation. Very simple. Now let us go into this flowchart. Now if you look into the flowchart, so primary meristems, I said they undergoes differentiation. So because of this differentiation process, you are getting dead cells like sclerenchyma, living cells like parenchyma as well as collenchyma. As I said earlier, now these living cells under certain conditions, again they become meristematic by a process called dedifferentiation. So because of this dedifferentiation, you are getting secondary meristem. This secondary meristem, once again, like we have examples for secondary meristems, I have given examples for secondary meristems also here in the flowchart, like interfascicular cambium of dicot stem, vascular cambium of dicot root, and cork cambium of dicot root and dicot stem. If you observe very carefully here, I have not given vascular cambium of dicot stem as such as example for the total secondary meristem. You know why? Because in dicot stems, vascular cambium is partially primary, partially secondary. How can you say? Because the vascular cambium of dicot stem is composed of interfascicular cambium and intrafascicular cambium. Intrafascicular cambium is a primary meristem. Interfascicular cambium is a secondary meristem. That's why while giving the examples for the secondary meristems, I have very clearly mentioned here interfascicular cambium of dicot stem. And whereas in case of dicot root, vascular cambium is completely secondary in origin, whereas in dicot stem, it is partially primary, partially secondary in origin. Whereas in dicot root, it is completely secondary in origin. Therefore, you can take the whole vascular cambium as an example for secondary meristem and as an example for the product of dedifferentiation. So that's very important. Now, when it comes to the cork cambium like Phyllogen, the cork cambium is completely secondary in origin both in dicot stem as well as dicot root. Now, this cambium, like whether it is vascular cambium or cork cambium, the cells of cambium redifferentiate. Once again they get matured, and that means once again they lose the division capacity and again become secondary meristematic. I mean permanent tissues. Like from the vascular cambium, you are getting secondary xylem and secondary phloem. From cork cambium, you are getting cork or phellum, as well as secondary cortex and phylloderm. This is very important. And one more important thing that we have to remember from this topic is, I said earlier, like in the beginning of this chapter, that growth is open form. Similarly, differentiation is also open form. How can you say? Because in the differentiation, like the same meristem, like tissues arising from the same meristem will have different kinds of structures at maturity. So, both growth as well as differentiations are open forms. This you have to remember. Now, what is development? It's another important aspect, though not much questions have appeared in the previous NEET, but in our internal exams many times the questions appeared in different ways, like by twisting even the questions have framed, and we cannot expect that in 2026 question will not appear. Like I think like the question may appear even from this topic in the 2026. So development is nothing but all that changes which happens from the germination of the seed till its senescence. Now, there is one important flowchart in the NCERT textbook itself about the whole process of development of a cell. One particular cell they are taking, not the total organism, the cell. So, meristematic cell after plasmatic growth again become meristematic by cell division, or it may differentiate and becomes mature, and after that it undergoes senescence and finally it enters into the death. So, this is one important aspect. And apart from this, plasticity is another important aspect that we have to understand in this topic. Plasticity is nothing but plants follow different pathways in response to the environment or phases of their growth. If they follow different pathway in response to the environment, then it is called environmental heterophyly. Whereas if they follow different pathways in response to the phases of its development, its own development, then it is called developmental heterophyly. In the NCERT book, they have also given different examples for environmental heterophyly and developmental heterophyly. For developmental heterophyly, we have examples like cotton, coriander, larkspur, all these you can take. Whereas for environmental heterophyly, we have one best example, that is buttercup. Buttercup is the best example. Buttercup scientifically called as ranunculus. And so we can conclude now that development is the sum of growth and differentiation. It is the sum of growth and differentiation. Now, another most important thing in this chapter, like most of the questions have appeared in previous NEETs. Even if you look from the 2015 onwards, most of the time questions have appeared from this particular topic, like plant growth regulators. So, in the plant growth regulators, we have different groups. There are growth promoters, growth inhibitors. There is one exception, that is ethylene. It has both growth promoting as well as growth inhibiting ability. Both are there. But mostly it acts like a growth inhibitor, though it has both growth promoting and inhibiting, but most of functions that it is possessing is growth inhibiting activities. Now, if you look the derivatives from which they have derived, like for example, Auxin, like indole 3-acetic acid, it is an indole derivative. Cytokinin, like kinetin, is an adenine derivative. Similarly, ABA, abscisic acid, it is a derivative of carotenoids. Gibberellins, gibberellic acid, it is a derivative of terpenes. Ethylene is a gaseous phytohormone. And one most important thing, most of the students that they do not keep their concentration about this sentence, that discovery of all these 5 hormones have happened accidentally. At the same time, most of them do not keep their concentration on one particular sentence of the discovery related to the auxin is their exact discovery, their exact site of production set by Charles Darwin and Francis Darwin. Charles Darwin and Francis Darwin have taken canary grass So the canary grass coleoptiles they have taken, and they found that the tips of coleoptile are the site of transmittable influence. I have highlighted here. You can keep underline in your textbook. If it is not underlined, it is most important. It is the site of transmittable influence. They did not say that it is auxin. They just said that it is a transmittable influence. And of course, they also proved through their experiment, with this experiment, that this transmittable influence have the influence in the phototropism aspect. And apart from that, the second important hormonal discovery, that is gibberellins discovery. If you look, gibberellins were discovered from a fungi, Gibberella fusicorae. This is a fungus, so this aspect is connected to the biological classification. This Gibberella fusicorae, remember, it is the perfect stage and it is belonging to Ascomycetes fungi. Though it is not mentioned in the NCERT, I think you might have come across in our internal exams about the group to which this particular fungi is belonging. It is belonging to Ascomycetes. It has an imperfect stage that itself is Fusarium moniliforme. Fusarium moniliforme. This fungus, this gibberellins have isolated. Similarly, Skoog and his co-workers have understood about effect of cytokinins through their experiments by taking internodal segments of tobacco plant. So they added auxins along with any one of the supplements like extracts of vascular tissues or yeast extract or even coconut milk or DNA. Then they found that these internodal segments of tobacco have become callous. If any one of these extracts were not supplied, and only when auxin alone is supplied, there was no callus production in the internodes of tobacco. So based on this, they found some cell division inducing substance in these components, and later it was Skoog and Miller have identified that it is cytokinin. And you all know that when it comes to the abscisic acid discovery, abscisic acid has been discovered by 3 different groups of scientists, and they have named it as inhibitor B, then abscissin 2, then dormin. And finally, they came to the conclusion that all these 3 are chemically similar and also structurally similar, and they gave a common name, that is abscisic acid. And when it comes to ethylene discovery, it was Cousins who identified that ethylene is a volatile gas that has the ability to cause ripening in the fruits. So this is what you can see here. Now let us go into the details of each hormone here. When it comes to the auxins, first isolated from human urine. Remember, auxins in 1928 itself, FW went isolated from coleoptiles of oat seedlings. But its chemical isolation was carried by isolating from human urine by Kogel and Smith. They are different. Of course, the names of these 2 scientists have not given the NCERT, but this is also very important thing. And now we have both natural auxins as well as synthetic auxins. When it comes to the natural auxins, we have 2 important natural auxins here, like IAA and IBA, and synthetic auxins like NAA, 2,4-D. These are all synthetic auxins. Even IPA is also there. Now, auxins have very important role in various aspects of plant growth and development. Like in case of pineapple, they promote flowering. In the same way, auxins also prevent fruit and leaf drop in the early stages, but promotes abscission of older leaves and fruits. Very, 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 very important. And they also have a very important role in apical dominance. Apical dominance. So, apical dominance is nothing but it is apical bud that suppresses growth of axillary buds. Thereby, plants growth linearly more than radially. Now, apart from that, we also have some like synthetic auxins 2,4-D that can be used as weedicide, or simply to say it can be used as a herbicide. And auxins also induces parthenocarpy. Auxins can also cause cell division. Auxins also involves in cell differentiation, like xylem differentiation. They have a role in cell division. They have a role in differentiation process also. This you have to remember that they play important role in xylem differentiation, not in the phloem differentiation, remember. Now let us go into the second important phytohormone, that is gibberellin. When it comes to the gibberellins, now we have more than 100 gibberellins, and most intensively studied form of the gibberellin is GA3. I have highlighted here. This you have to remember. Most intensively, more than 100 gibberellins are there. Among them, most intensively studied form is GA3. And gibberellins have so many important aspects, like they help, they improve the fruit shape and length of the fruit in case of apple. They delay fruit senescence. This is very, 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 very important. Fruit senescence, not the leaf senescence. And apart from that, they are also useful in brewing industry, and in sugarcane crop they can also increase the yield by increasing the stem elongation. So gibberellins can remove genetic dwarfism because they have the ability to cause cell elongation, not cell differentiation. Auxin involves in xylem differentiation, whereas gibberellins involves in cell elongation. So they play important role in elongation phase, not in the maturation phase. Understood? Whereas auxins have a role in meristematic phase as well as in differentiation phase. So that is the importance of this. Now spraying gibberellins can also hastens maturity period in case of conifers, and apart from that they also play important role in bolting process. When it comes to the cytokinins, kinetin, it is a degradative product of DNA. It does not occur naturally in the plant. This is most important line in the cytokinin topic. It does not occur naturally in the plants. That is the importance of this kinetin. Later, natural cytokinins have also identified one of the natural cytokinin is geatin. And these cytokinins have so many important roles in the whole process of plant growth and development, like of course they are synthesized in cells actively undergoing cell division, and they play important role in the production of new leaves as well as chloroplast in leaves. And apart from that, lateral shoot growth, adventitious shoot growth, not adventitious root growth. Remember, adventitious root formation was initiated by auxins. Auxins initiate root formation from the stem cuttings in horticulture, whereas cytokinins causes adventitious shoot formation, not root formation. Don't get confused. So this is what, these are some tricky lines in the NCERT where actually like most of the students are getting confused. So another important thing, they also promote nutrient mobilization and delay the leaf senescence. Gibberellins delay fruit senescence. Cytokinin delays leaf senescence. So there is a difference here. So you have to be very particular for remembering these components, because this total chapter is memory based and you have to be very specific and particular in remembering some of the important aspects. So, gibberellins delays fruit senescence, whereas cytokinins delays leaf senescence. Now, let us come to the ethylene. When it comes to the ethylene, I already told it was discovered in relation to the fruit ripening process. It plays a very important role in senescence and abscission. So, it has antagonistic effect to the cytokinins. Cytokinins promotes, delays senescence, whereas ethylene promotes senescence of leaves as well as abscission of leaves and flowers. And of course, ethylene, like it induces, it has very important influence in horizontal growth of seedlings, swelling of the axis, as well as apical hook formation. So, these are all some important things we have to know. And apart from that, I also told you that it is fruit ripening hormone, and during the ripening process, it increases the rate of respiration. So, this phenomena itself is called respiratory climactic. Respiratory climactic. Now, apart from this ethylene also has a very important role in breaking of seed and bud dormancy, and it also initiates the seed germination. Remember, there are two hormones which have a role in the germination of the seeds. Gibberellins can also promote seed germination. Ethylene can also promote seed germination. But ethylene is promoting seed germination in peanut seeds, not in every plant. In most of the cases, like ethylene plays an important role in breaking seed and bud dormancy. But it is specifically playing a very important role in germination of peanut seeds. So, ethylene and gibberellin both have a role in the process of seed germination. Now, ethylene promotes rapid internodal elongation, particularly in deep water rice plants. And apart from that, it also increases root growth, root hair formation, like it increases surface area of the root and root hair formation. So, root hair formation it is increasing, that you have to remember. And ethylene is also used in initiating flowering and synchronized fruit set in case of pineapple. So, in pineapple, this flowering process is influenced by both auxins as well as ethylene. This is very, very, very important. In addition to this, ethylene, as it is in gaseous form, now we have a chemical powdery form of ethylene, that itself is ethafone. So, this ethafone, it hastens the ripening process of tomato, apple, as well as accelerates abscission of flowers and fruits, like particularly thinning of cotton, walnut, cherry, and it also promotes female flower production in case of cucurbits. And you have to remember here, gibberellins delaying fruit senescence in apple, but ethylene is promoting fruit senescence in apple. So, both are antagonistic. Now, lastly, let us see about the abscisic acid. Abscisic acid is playing a very important role or regulating about the dormancy and abscission process. You have to be very careful here. Ethylene has a role in abscission and senescence, whereas ABA has its role in abscission process but not in the senescence. This you have to remember. And of course, ABA promotes dormancy in the seeds, like it inhibits the seed germination, and it is also called a stress hormone. Why? Because this ABA under stress condition, like water stress conditions or various, it makes the plant to tolerate various environmental stresses by closing the stomata. Here you have to remember, cytokinins can promote opening of stomata, whereas ABA promotes closure of the stomata. particularly under water stress condition. That's why it is called a stress hormone. And it is also playing a very important role in seed development, maturation and dormancy. And in most situations, this ABA is antagonistic to gibberellins. This is a very important line from this particular topic. And ABA is also playing, like, helps the seeds to withstand desiccation and other factors unfavorable for plant growth. So these are the important aspects with respect to the plant growth regulators. Now let us see the common mistakes that most of the students do during examination. Now most of the students are getting confused between arithmetic growth and geometric growth. As I said, arithmetic growth represents linear increase at a constant rate. Constant rate, this you have to remember. Whereas geometric growth represents exponential increase of weight or cell number or height, etc. So in both you can see the height. Arithmetic growth can help in height. Geometric growth can also help in height. But what makes the difference? Here the increasement in the height, like here in the sense arithmetic growth, increasement in the height is taking place at a constant rate. But in geometric growth, it is taking place at exponential rate. This difference you have to remember. And the second important aspect where most of the students are getting confused is difference between dedifferentiation, differentiation, dedifferentiation and redifferentiation. I very clearly gave all the details of various definitions, like how to remember the definitions of these three aspects. So try to recollect it and go through your textbook once again so that you will get the clarity. Now another important thing that most of the students gets confused is gibberellins delay the fruit senescence, whereas cytokinins delays leaf senescence. Both are different. Gibberellins cannot delay leaf senescence and cytokinins cannot delay fruit senescence. Delay of leaf senescence by cytokinins by mobilizing nutrients is called Richmond-Lang effect. Remember, what is it? Richmond-Lang effect. That is delay of leaf senescence by cytokinins. Now, many of them are also getting confused among antagonistic roles of different phytohormones. I have told you, like, where different hormones and their antagonistic effect. Thus, that you try to recollect once again. Then another important thing, ethylene promotes senescence and abscission, whereas ABA accelerates abscission, not the senescence. This also you have to remember. And in most situations, this is an NCERT line, in most situations, ABA is antagonistic to GAs. ABA is antagonistic to GAs. So this is very important. Now let us see interconnections of this chapter with other chapter. Now many aspects of this chapter are directly or indirectly connected with anatomy of flowering plants. Let us take some examples. Plant growth is generally indeterminate. Under this we are discussing about meristems, various types of meristems. So meristems we have come across under anatomy of flowering plants. Similarly, differentiation, dedifferentiation and redifferentiation. These topics also studied under anatomy of flowering plants during secondary growth. In secondary growth, you are getting all these aspects. So if you are thorough this particular chapter, it becomes very easy to remember plant growth and development. Apart from that, we also have some interlinks with biomolecules. Let me tell. For example, take kinetin. What is this kinetin? Kinetin is a derivative of adenine. It is a derivative of adenine. Adenine is a purine. Adenine is a purine. Purine, adenine and guanine, both are purine. So here it is an adenine derivative. It's a nitrogen base. So we come across in biomolecules chapter. Similarly, ABA is a derivative of carotenoids. Where did you come across carotenoids? Carotenoids we studied in cell unit of life under chloroplast. We also studied in biomolecules under secondary metabolites. We also studied in photosynthesis during the whole process of photosynthesis. So, carotenoids. And apart from that, gibberellins are terpenoids. They are terpenoid compounds. So, where did you come across it? It is all again came in biomolecules under the subtopic secondary metabolites. Have you got the clarity? So, this is very important. Next, another important thing, if you look the discovery of cytokinin in the very introduction of plant growth regulators, discovery of cytokinin, where we have studied about callus formation, how the callus is produced from internodes of tobacco. So, this information is linked with tissue culture. So, these are the some tissue culture of plant biotechnology applications. So, these are the some areas of this chapter that have interconnection with other chapters. Now, before we close, let us quickly remember some of the important aspect. Like let us take one important definition of this chapter. I am again giving the definitions of differentiation, dedifferentiation, redifferentiation. This you have to remember very carefully. And the second one, important lines of the NCERT. Of course, every line of the NCERT is very important. You cannot avoid any line of the NCERT. But what I am feeling is these two are very important and critical because most of the students, they don't remember particularly these two lines. Both growth and differentiation are open forms, because I have seen that many of the students, like whenever the question is asked, they are thinking and remembering only open form is concerned to the growth but not to the differentiation. But it is linked with both growth as well as differentiation are open forms. This is very important line. And the second thing, development is the sum of growth and differentiation. These two things you have to remember. Now, one important diagram. Of course, every diagram is important in the NCERT book. You cannot avoid any diagram. I feel this is most important because many of the students are getting confused and they are not properly understood about this diagram. This diagram is related to Charles Darwin's phototropic experiment by taking canary grass. See, in this diagram, when unilateral light is exposed to the coleoptile, there was a bending. When coleoptile tip was removed and exposed, there was no growth. When coleoptile tip is covered with light tight cap or opaque, there was no curvature. Whereas when coleoptile tip is covered with transparent cap, there was a curvature. Here there was no curvature, here there is a curvature. Understood? So, this is one most important diagram. And one most important exception of this chapter, you can take like ethylene could fit under both growth promoter as well as growth inhibitor. So, this is another important thing we have to know. And common trap, most of the students making mistake about understanding about this particular line of NCERT. What is that? Auxins help to prevent leaf and fruit drop in the early stages, but abscission of older and mature leaves and fruits, abscission of older and mature leaves. That means in the early stages, it is preventing leaf and fruit drop, but in the later stages, it is promoting leaf and fruit drop. It has both. This is very important. And apart from that, this is also another important thing, like cytokinins delays leaf senescence, whereas gibberellins delays fruit senescence. Gibberellins delays fruit senescence. So, this is another important aspect of this chapter. Now, let us see some of the objective questions from Plant Growth Development chapter. Now, here we have some important questions from this chapter. So, let me take this assertion and reason. Ethylene is largely inhibitor of growth activities in plants. It is true statement. It is a direct statement from the NCERT. Let me see the reason. Ethylene promotes senescence and abscission of plant organs. Why ethylene is called mainly as growth inhibitor? Because it is promoting senescence and abscission. As they gave the correct explanation, now it becomes first option, like assertion and reason is true and R explains here. Similarly, let me see some other questions also, like let us take this statement type of question. A differentiated cell may redifferentiate and then dedifferentiate. A differentiated cell may redifferentiate and then dedifferentiate. Is it correct? Differentiated cell, does it undergoes dedifferentiation or redifferentiation? It undergoes this dedifferentiated cell, then undergoes redifferentiation. So this statement becomes false. Statement A is false. Now statement 2, differentiation results in the development of structures that is commensurate with function of the cells finally has to perform. It is true. It's a direct line. So here statement 1 is false, but statement 2 is true. So similarly, there are many questions, like most probably, as we observed in the beginning, most of the questions have come from the plant growth regulators, but there is also chance for getting even from other chapters. So look, every question, every aspect of the line textbook, like every line of the NCERT and every diagram of the NCERT, you must follow thoroughly before going to the exam. Now, so this is about plant growth regulators. Thank you very much.